Hi everyone, my name is Justine Y Hansen. I'm from McGill University, and the title of this presentation is Mapping Neurotransmitter Systems to the Structural and Functional Organization of the Human Neocortex. This is corresponding to poster WTH780. So the idea here is that we know that these receptors, which are proteins embedded within the cell membrane, we know that when a neurotransmitter binds to it, the configuration of either the receptor itself, or if it's a metabotropic receptor, then a downstream ion channel, it's going to change, allowing ions to flow in or out of the cell. And this is going to change the electrical potential within the cell, which is going to affect whether or not this neuron is going to fire an action potential. So this makes receptors key modulators of firing rate, and therefore also brain connectivity, so whether or not the two neurons are going to synapse onto one another, and dynamics, the firing rate of the cell. And what we wanted to do was see if we can find this relationship not at the level of the single cell, but at the level of the whole cortex and across multiple neurotransmitter systems. And neurotransmitter receptors, because they are modulating both brain connectivity and dynamics, this makes them key players within the relationship of brain structure and function. So we really wanted to assess the relationships of neurotransmitter systems along the brain structural and functional organization. So to do this, we collected a bunch of PET tracer image data across 19 different neurotransmitter receptors and transporters, 9 neurotransmitter systems, and 1,238 healthy participants. So this data is group average, is coming from different studies, different research groups, and we compile it into one single atlas that is available at my GitHub, but also on the Python software toolbox called NeurMaps. And what we do with our analyses is that we have this we conduct them using this PET atlas, but then we replicate them using an autoradiography dataset from Zillis and Nicola Palomero Gallagher in 2017. But one of the first things we do with this data is that our first main conclusion is that the receptor gradient, we find a receptor gradient that separates classes of laminar differentiation and is associated with synapse density. So I'll go into that a little bit slower. So if you imagine that every brain region, there is this fingerprint of receptors. So for the 19 receptors that I have in this atlas, there's a density for each one, and this will comprise the fingerprint, the receptor fingerprint of a specific brain region. And so for every pair of brain regions, if I correlate those two fingerprints, I get a region by region matrix of receptor similarity. This is indexing how similar two brain regions are in terms of their receptor fingerprint, but more conceptually, it's indexing how similar two brain regions might respond to the same neural signal. And here I'm showing the distribution of receptor similarity and also that it decays exponentially with distance. So one of the things we do with this receptor similarity matrix is that we calculate the first principal component, or this gradient of receptor similarity, and we find that it separates these classes of laminar differentiation. This is coming from Markle Mesolum, so paralimbic classes from idiotypic classes, and we find that this gradient is correlated with synapse density, which means that the primary axis of variation of receptor density is actually related to whether or not a brain region has a lot of synapses. Our second main conclusion is that neurotransmitter systems are systematically aligned with structural and functional connectivity. So this conclusion is coming from comparing that receptor similarity matrix to structural connectivity and functional connectivity. These are ordered by the Yo-Crenin 2011 resting state networks. And we find that receptor similarity is greater when brain regions are connected with one another and greater when brain regions are within the same functional network. We also find that receptor similarity is correlated with both structural connectivity and functional connectivity, and this is after regressing distance from both of these matrices. The next conclusion that we make is that neurotransmitter receptor profiles, especially ionotropic receptors, shape oscillatory neural dynamics. So this is coming from looking at MEG-derived power distributions along the cortex. So this is coming from HTP. I have these 6 different power bands, and I'm using multilinear regression models to fit the receptors as input variables onto these power bands as output variables. And I find my main conclusion here is that the fit is very high. So I find that receptors map quite closely onto these power distributions from MEG. But then I use this technique called dominance analysis to distribute the fit of this model across the input variables. So this has given me an idea of how much each of these input variables, in my case receptors, are contributing to my to the high fit between receptors and power dynamics. We replicate this in the autoradiography dataset, and especially here, also in the PET dataset, but especially here because it's a different set of receptors, we find that the most contributing receptors are actually ionotropic. So AMPA and MDA, these are all ionotropic glutamate receptors, we have GABA, alpha-4 beta-2, and this kind of makes sense because my ionotropic receptors are the ones that are responding more immediately to the binding of the neurotransmitter, whereas the metabotropic receptors are those that are more modulating the excitability of a neuron or kind of tuning its gain. The next conclusion I have from this project is that we find that microarray gene expression can generally not be used as a proxy for neurotransmitter receptor density. So where is this coming from? So I have these this PET data, and before this was made available, it was kind of hard to come across, especially multi-system PET receptor data. And so a lot of the time trying to make conclusions about receptor densities, instead of using the receptor densities themselves from something like autoradiography or PET, researchers would use gene expression as a proxy under the assumption that the two are correlated. And this is totally fair considering it was kind of hard to get your hands on density data. But now that I had this PET receptor density data, I wanted to just compare the two and see, is it the case that we can make this assumption? And so I'm using microarray gene expression data from the Allen Human Brain Atlas, and I'm finding that in most cases the two aren't actually correlated. So the blue scatter plots are those where there is no significant correlation, and the yellow scatter plots are those where there is a significant correlation. We looked a little bit further into why this might be the case, and we find that low expression density correspondence is driven by low intersubject variability. So here I'm showing a correlation between the differential stability of a gene, so that's how variable it is across the 6 donor brains in the Allen Human Brain Atlas, and the correlation between the gene expression and receptor density. And I'm finding that genes that are very stable tend to be highly correlated with their receptors. So this seems to suggest that this relationship has something to do with individual variability. Our final conclusion is that we find a topographic gradient of receptor profiles that links monoamine receptors with effective psychological processes. So again, I'm going to go through that a little slower. What we do is we do a partial least squares analysis between the receptor atlas and Neurosynth-derived meta-analytic cognitive activation. So here I'm showing there's one significantly latent variable that accounts for about 55% of the variance explained between the two data sets. This is against a spin null. And this extracts a topographic pattern of receptor densities and also cognitive activation patterns that's really linking these receptors with different cognitive processes. And we cross-validate this model and specifically find that the receptors that contribute a lot to this relationship tend to be monoamine metabotropic receptors. So these are especially serotonergic and dopaminergic receptors and transporters. And that the cognitive terms or the cognitive processes that are aligned with these receptors tend to be related to emotion processing. So things like emotion, valence, fear, emotion regulation. So we're really finding a link between these mood-related receptors, emotion processing, and that follows this topographic distribution that I'm showing here in panel B and C. So this brings me to the end of my presentation. We collect a bunch of PET receptor maps and we make them available, so please go ahead and use them in your own research. We situate chemoarchitecture within this multi-scale organization of the brain. We find that neurotransmitter receptor profiles closely align with the structural connectivity of the brain, and mediate is linked with function. And finally, the overlapping topographic distributions of these receptors ultimately manifest as patterns of cognitive specialization. So the code and data is on GitHub, and it's also on this Python software toolbox called Neuromaps, which is poster WTH813, so I'm also presenting on that one. Thank you so much. This is my lab. The first paper to do with the receptor distributions, so most of what I presented here is this preprint, but there's also the second preprint, which is about the gene expression to the receptor densities that I presented a little bit about. And yeah, the institution and funding. Thank you very much.